Senescence. Senescence refers to the process by which cells, tissues, and organisms experience gradual deterioration and function as they age. In cellular biology, cellular senescence is a permanent cell cycle arrest that occurs in response to various stressors, including DNA damage, oxidative stress, telomere shortening, and oncoprotein activation. This mechanism is thought to act as a protective mechanism to prevent the proliferation of damaged or potentially cancerous cells, but it also contributes to aging and age-related diseases. Senescence is a critical biological phenomenon observed in multicellular organisms and is categorized into different types: replicative senescence, stress-induced premature senescence, SIPS, and senescence in response to damage. The ultimate consequence of cellular senescence is the cessation of cell division, but the senescent cell remains metabolically active and secretes factors that can contribute to aging and the development of age-related diseases. The ultimate consequence of cellular senescence is the cessation of cell division, but the senescent cell remains metabolically active and secretes factors that can contribute to aging and the development of age-related diseases. 1. Mechanisms of cellular senescence. Cellular senescence is a complex process that involves several molecular and biochemical pathways. The main features of senescence are cell cycle arrest, altered gene expression, and the senescence-associated secretory phenotype (SASP). Senescence can be triggered by various intrinsic and extrinsic factors. 1.1 DNA damage response in cell cycle arrest. The DNA damage response (DDR) plays a central role in the induction of senescence. In response to DNA damage caused by factors like oxidative stress, telomere shortening, or genotoxic stress, the cell activates several key tumor suppressor proteins to arrest the cell cycle and prevent the propagation of damaged DNA. The most important tumor suppressors involved in this process are p53. The transcription factor p53 plays a central role in the DNA damage response by inducing cell cycle arrest in the G1 phase. It triggers the expression of p21, which inhibits cyclin-dependent kinases CDKs and prevents progression through the cell cycle. P1604A, another critical player in senescence. P1604 inhibits cyclin-dependent kinases CDKs and prevents the phosphorylation of retinoblastoma protein RB, which in turn leads to cell cycle arrest in the G1 phase. These mechanisms ensure that cells with DNA damage do not continue to proliferate, thus preventing the propagation of mutations. 1.2 Telomere shortening in senescence. Telomeres are repetitive DNA sequences located at the ends of chromosomes that protect the genome from degradation and prevent chromosome fusion. However, with each cell division, telomeres shorten, and when they reach a critically short length, the cell enters a state of replicative senescence. This is particularly important in somatic cells that lack the telomerase enzyme, which normally replenishes telomeres in germ cells and certain stem cells. Telomere theory of aging. The progressive shortening of telomeres over time is believed to be one of the primary contributors to cellular senescence and aging. When telomeres shorten beyond a critical threshold, they trigger a DNA damage response that leads to cell cycle arrest and senescence. 1.3 Oncogene-induced senescence (OIS). Oncogenes such as ROS, MYC, and BCR-ABL can also induce senescence in response to their overexpression. This phenomenon, known as oncogene-induced senescence (OIS), serves as a protective mechanism against uncontrolled cell proliferation and cancer formation. The activation of these oncogenes leads to DNA damage and the activation of the p53, p21, and p1604A RB pathways, resulting in cell cycle arrest and senescence. 1.4 Oxidative stress and senescence. Oxidative stress, caused by an imbalance between the production of reactive oxygen species, ROS, and the ability of the cell to neutralize them, is a significant driver of senescence. ROS can damage cellular components such as lipids, proteins, and DNA, leading to a breakdown in cellular function. The accumulation of oxidative damage over time is thought to contribute to both cellular and organismal aging. ROS and mitochondria. Mitochondria are a major source of ROS due to the electron transport chain. As cells age, mitochondrial function declines, leading to increased ROS production, which accelerates senescence. 2. Senescence-associated secretory phenotype (SASP). Senescent cells, although no longer proliferating, remain metabolically active and secrete a variety of bioactive molecules, collectively known as the senescence-associated secretory phenotype (SASP). These molecules include pro-inflammatory cytokines, chemokines, growth factors, and matrix metalloproteinases (MMPs). The SASP can have both beneficial and detrimental effects on tissue homeostasis. Inflammation. The pro-inflammatory cytokines secreted by senescent cells contribute to chronic low-grade inflammation, which is a hallmark of aging and age-related diseases. Tissue remodeling. The secretion of MMPs can lead to tissue remodeling and fibrosis, impairing the function of various organs. Immune modulation. While the SASP may aid in clearing damaged cells, chronic secretion of these factors can lead to immune evasion and promote the development of age-related diseases such as cancer. 3. Role of senescence in aging and disease. Cellular senescence plays a crucial role in both aging and age-related diseases. While senescence prevents the proliferation of damaged cells and acts as a tumor suppressive mechanism, the accumulation of senescent cells in tissues can impair normal tissue function and regeneration, leading to the aging process. 3.1 Aging. As an organism ages, the accumulation of senescent cells increases in various tissues, including the skin, muscle, liver, and kidney. The presence of these senescent cells contributes to the decline in organ function, impaired tissue repair, and the onset of age-related diseases. 3.2 Age-related diseases. Senescent cells have been implicated in the pathogenesis of several age-related diseases, including cancer. The accumulation of senescent cells and the inflammation caused by the SASP may promote the development of tumors. Neurodegenerative diseases. In conditions like Alzheimer's disease and Parkinson's disease, senescence-related inflammation and cellular dysfunction contribute to neuronal loss and cognitive decline. Osteoarthritis. Senescent cells accumulate in the synovium and articular cartilage, contributing to inflammation and tissue degradation in osteoarthritis. 4. Targeting senescence for therapeutic purposes. Given the role of senescence in aging and disease, therapeutic strategies aimed at targeting senescent cells have been explored. These strategies include senolytics, drugs that selectively eliminate senescent cells, thereby alleviating the deleterious effects of their accumulation. Examples include dasatinib and quercetin. Senolytics, drugs that selectively eliminate senescent cells, thereby alleviating the deleterious effects of their accumulation. Examples include dasatinib and quercetin. Senomorphics, compounds that modulate the SASP to reduce its harmful effects without eliminating senescent cells. Previous year CSIR-NET questions. Senescence CSIR-NET June 2017. Question: which of the following proteins is primarily involved in inducing cell cycle arrest during cellular senescence? Acyclin D. BP53CCMYCDRB. Answer: BP53. P53 is a key regulator of cell cycle arrest in response to DNA damage, promoting senescence. CSIR-NET December 2019. Question: the process by which the telomeres shorten with each cell division and trigger cellular senescence is called oncogene-induced senescence. B. Replicative senescence. C. Senescence-associated secretory phenotype. D. Mitochondrial dysfunction. Answer: B. Replicative senescence. Replicative senescence occurs due to telomere shortening, causing cell cycle arrest. CSIR-NET June 2020. Question: which of the following molecules is most likely to be secreted by senescent cells and is associated with inflammation? AP21. BP1604A CIL6 D cyclin E. Answer: CIL6. IL6 is a pro-inflammatory cytokine that is secreted by senescent cells as part of the SASP. This lecture on senescence provides detailed insights into the biological mechanisms, molecular pathways, and implications of senescence in aging and disease. It also includes previous year questions to guide your exam preparation and ensure success in the CSIR-NET JRF Life Sciences exam. You said. Program cell death. Aging and senescence important points. ChatGPT said. Here's an elaborate lecture on programmed cell death, aging, and senescence, focused on the key points relevant to CSIR-NET JRF Life Sciences. This lecture provides a comprehensive understanding, ensuring you are prepared with crucial exam-oriented details. It also includes 2 to 3 previous year questions with answers for further reinforcement. Programmed cell death (PCD). Programmed cell death (PCD) refers to a regulated process in which cells undergo a controlled death to maintain tissue homeostasis or eliminate damaged or dangerous cells. PCD is distinct from necrosis, which is an uncontrolled, traumatic form of cell death. There are 3 main types of PCD in multicellular organisms. 1.1 Types of programmed cell death. Apoptosis. Apoptosis is the most well-known form of PCD. It is a highly regulated process that ensures the removal of damaged or unwanted cells without causing an inflammatory response. Apoptosis is characterized by cell shrinkage, nuclear fragmentation, chromatin condensation, and membrane blebing. Key regulators. Caspases, proteases that play a central role in apoptosis by cleaving cellular proteins and triggering the dismantling of the cell. BCL2 family proteins regulate mitochondrial outer membrane permeabilization, which is crucial for the initiation of apoptosis. Cytokines modulate apoptotic pathways and inflammation, influencing immune responses. Autophagic cell death. Autophagy refers to the process of degrading and recycling cellular components. Under certain conditions, it can lead to cell death, especially in cases of severe stress. Unlike apoptosis, which leads to the breakdown of the entire cell, autophagic cell death often results in partial degradation. Necroptosis. Necroptosis is a regulated form of necrosis that shares features with both apoptosis and necrosis. It occurs when caspase-dependent apoptosis pathways are inhibited, and a series of It is a highly regulated process. Programmed cell death (PCD). Programmed cell death (PCD) refers to a regulated process in which cells undergo a controlled death to maintain tissue homeostasis or eliminate damaged or dangerous cells. PCD is distinct from necrosis, which is an uncontrolled, traumatic form of cell death. There are 3 main types of PCD in multicellular organisms. 1.1 Types of programmed cell death. Apoptosis. Apoptosis is the most well-known form of PCD. It is a highly regulated process that ensures the removal of damaged or unwanted cells without causing an inflammatory response. Apoptosis is characterized by cell shrinkage, nuclear fragmentation, chromatin condensation, and membrane blebing. Key regulators. Caspases, proteases that play a central role in apoptosis by cleaving cellular proteins and triggering the dismantling of the cell. BCL2 family proteins regulate mitochondrial outer membrane permeabilization, which is crucial for the initiation of apoptosis. Cytokines modulate apoptotic pathways and inflammation, influencing immune responses. Autophagic cell death. Autophagy refers to the process of degrading and recycling cellular components. Under certain conditions, it can lead to cell death, especially in cases of severe stress. Unlike apoptosis, which leads to the breakdown of the entire cell, autophagic cell death often results in partial degradation. Necroptosis. Necroptosis is a regulated form of necrosis that shares features with both apoptosis and necrosis. It occurs when caspase-dependent apoptosis pathways are inhibited, and a series of signaling pathways, including receptor-interacting protein kinase 3 (RIPK3), activate necrotic cell death. 1.2 Mechanisms of programmed cell death. PCD is mediated by a variety of signaling pathways, often involving the activation of death receptors, intracellular signaling molecules, and mitochondrial events. Intrinsic pathway. Mitochondrial pathway involves the release of cytochrome C from the mitochondria into the cytoplasm, leading to caspase activation. Extrinsic pathway. Death receptor pathway involves the activation of cell surface death receptors, such as FOS or TNF receptors, which recruit and activate caspases. Autophagic pathway. The LC3 protein and BDCL-IN1 are critical in initiating autophagy, while mTOR regulates autophagic activity under nutrient-sensing conditions. 1.3 Functions of programmed cell death. Development. PCD is crucial for shaping organs and tissues during embryonic development, such as the removal of excess neurons or digit separation. Homeostasis. PCD removes damaged or infected cells, maintaining tissue integrity. Immune response. During immune responses, apoptosis helps eliminate infected or transformed cells, preventing the spread of infection or cancer. Aging. Aging refers to the gradual decline of an organism's functional capacity over time, typically involving a complex interplay of genetic, environmental, and cellular factors. It results in a decrease in biological functions and an increased susceptibility to disease. 2.1 Mechanisms of aging. Genetic program theory. The aging process is partially controlled by genetic programs that regulate the lifespan of organisms. This theory suggests that the genes regulating growth, reproduction, and development also control the rate of aging. Damage accumulation theory. This theory proposes that aging results from the accumulation of genetic and molecular damage over time, due to factors such as oxidative stress, radiation, and metabolic byproducts. This damage leads to functional decline in cells, tissues, and organs. Free radicals. Overproduction of reactive oxygen species, ROS, contributes to oxidative damage, leading to aging. DNA damage. Accumulation of mutations in mitochondrial and nuclear DNA impairs cellular functions. Telomere shortening. Telomeres, protective caps at the end of chromosomes, shorten with each cell division. When telomeres become critically short, cells enter senescence or undergo apoptosis, contributing to the aging process. Mitochondrial dysfunction. As mitochondria age, their ability to generate ATP decreases, leading to reduced cellular energy. The accumulation of mitochondrial mutations also contributes to aging-related diseases, such as neurodegeneration. 2.2 Aging at the cellular level. Cellular senescence. As cells age, they accumulate damage and enter a state of irreversible cell cycle arrest known as senescence. Stem cell exhaustion. Aging leads to the depletion of stem cells, which results in impaired tissue regeneration and function. Inflammation. Chronic low-grade inflammation, or inflammaging, contributes to aging and age-related diseases. Senescence. Cellular senescence is a state in which a cell permanently exits the cell cycle due to DNA damage, oxidative stress, or telomere shortening. Senescence is considered a double-edged sword. While it prevents the proliferation of damaged cells, it also contributes to aging and age-related diseases. 3.1 Molecular mechanisms of senescence. P53 and P21. The tumor suppressor P53 activates the expression of the cyclin-dependent kinase inhibitor P21, leading to G1 arrest. P1604A and RB. P1604 inhibits the cyclin-dependent kinase 4, CDK4, leading to hyperphosphorylation of RB and a block in cell cycle progression. Telomere shortening. Telomere shortening activates the P53 pathway, inducing senescence as a protective mechanism against DNA damage. 3.2 Senescence-associated secretory phenotype, SASP. Senescent cells secrete various pro-inflammatory cytokines, chemokines, growth factors, and matrix metalloproteinases, MMPs, collectively known as the SASP. The SASP has both beneficial and detrimental effects. Positive role, helps in tissue repair and wound healing. Negative role, promotes chronic inflammation, fibrosis, and the progression of age-related diseases. 3.3 Role of senescence in aging and disease. Aging. The accumulation of senescent cells in tissues contributes to the decline in organ function and the deterioration of regenerative capacity. Cancer. Senescent cells can contribute to cancer progression through the SASP, which may create a microenvironment conducive to tumor growth. Neurodegenerative diseases. Senescence in neural cells contributes to neuronal dysfunction and cognitive decline in diseases like Alzheimer's and Parkinson's. Osteoarthritis. Senescent cells accumulate in joint tissues, causing inflammation and tissue degradation. Previous year CSIR-NET questions. CSIR-NET June 2018. Question. Which of the following proteins is most involved in inducing apoptosis by mitochondrial pathway? A. CASPASE3. B. Cytochrome CCP53. D. BCL2. Answer. B. Cytochrome C. Cytochrome C released from mitochondria is a critical step in the intrinsic apoptotic pathway. CSIR-NET December 2020. Question. Aging is most strongly associated with the accumulation of which of the following? A. Telomerase. B. Reactive oxygen species C. Cyclins. D. Growth factors. Answer. B. Reactive oxygen species. ROS accumulation leads to oxidative damage and contributes significantly to aging. CSIR-NET June 2017. Question. The accumulation of senescent cells leads to which of the following conditions? A. Tumor suppression. B. Chronic inflammation. C. Reduced inflammation. D. Cell proliferation. Answer. B. Chronic inflammation. The SASP secreted by senescent cells promotes chronic low-grade inflammation. This lecture encapsulates the essential points of programmed cell death, aging, and senescence, providing a highly detailed, exam-focused understanding. The previous year questions have been included to ensure that you're thoroughly prepared for the CSIR NET JRF Life Sciences exam. You said. System physiology, plant photosynthesis. ChatGPT said. Here's an elaborate lecture on photosynthesis, focusing on key concepts and exam-oriented details to help you prepare for the CSIR NET JRF Life Sciences exam. This lecture includes essential information from base to advanced level, without any wasted space, with 2 to 3 previous year questions and answers tailored specifically for the exam. Photosynthesis in plants. Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy into chemical energy in the form of glucose and other organic molecules. This process is vital for life on Earth as it serves as the primary source of energy for almost all living organisms, directly or indirectly. It occurs in the chloroplasts of plant cells, primarily within the thylakoid membranes and stroma. Key components involved in photosynthesis: chloroplasts. Chloroplasts are the organelles where photosynthesis occurs. They contain chlorophyll, the green pigment that absorbs light energy. Chloroplasts are equipped with thylakoids, where the light reactions of photosynthesis occur, and the stroma, the site for the Calvin cycle dark reactions. Chlorophyll. Chloroplasts are equipped with thylakoids, where the light reactions of photosynthesis occur, and the stroma, the site for the Calvin cycle dark reactions. Chlorophyll. Chloroplasts are equipped with thylakoids, where the light reactions of photosynthesis occur, and the stroma, the site for the Calvin cycle dark reactions. Chlorophyll. Chlorophyll absorbs light, primarily in the blue 430-450 nanometers and red 640-680 nanometers regions of the spectrum, and reflects green light, giving plants their characteristic color. There are several types of chlorophyll, such as chlorophyll A, chlorophyll B, and carotenoids, which play roles in light absorption and protection. Light energy. Light energy is harnessed by chlorophyll and other pigments, exciting electrons that initiate the photosynthetic process. Photosynthesis stages. Photosynthesis occurs in 2 main stages: light-dependent reactions, also called light reactions, and light-independent reactions, also known as the Calvin cycle or dark reactions. 1. Light-dependent reactions. These reactions occur in the thylakoid membranes and require light to occur. The primary purpose is to convert light energy into chemical energy in the form of ATP and NADPH. Photon absorption. Photons of light are absorbed by chlorophyll and other pigments. This energy excites electrons to a higher energy state. Water splitting photolysis. The excited electrons are transferred through a series of electron carriers, known as the electron transport chain, etc. Water molecules are split to replenish the lost electrons in chlorophyll, producing oxygen as a byproduct and releasing protons into the thylakoid lumen. This process is known as photolysis. ATP and NADPH formation. As electrons travel through the electron transport chain, ATP is synthesized via chemiosmosis through ATP synthase as protons flow back into the stroma. The electrons are finally transferred to NADP+, reducing it to NADPH, which will be used in the Calvin cycle. 2. Light-independent reactions. Calvin cycle. The Calvin cycle takes place in the stroma of the chloroplast and does not require light directly. The main goal is to convert carbon dioxide into organic molecules like glucose using the energy from ATP and NADPH produced in the light reactions. Carbon fixation. The enzyme Rubisco fixes carbon dioxide (CO) from the atmosphere by attaching it to ribulose bisphosphate, RUB, a 5-carbon sugar. This forms a short-lived 6-carbon compound that quickly breaks down into 2 molecules of 3-phosphoglycerate, 3-PGA. Reduction phase. ATP and NADPH generated in the light reactions are used to convert 3-PGA into glyceraldehyde, 3-phosphate, G3P, a 3-carbon sugar. G3P is a molecule that can be used to form glucose and other carbohydrates. Regeneration of RUB. Some of the G3P molecules are used to regenerate RUB, ensuring that the cycle can continue. This requires the consumption of additional ATP. Production of glucose. The remaining G3P molecules are used to form glucose and other carbohydrates, which serve as an energy source for the plant and other organisms. Factors affecting photosynthesis: light intensity. Photosynthesis increases with light intensity until the plant reaches a point of saturation, where increasing light further has no effect. Carbon dioxide concentration. The rate of photosynthesis is directly proportional to the concentration of CO up to a certain point. Beyond that, increasing CO will not further increase the rate. Temperature. Photosynthesis is temperature dependent. At high temperatures, the enzymes involved in the Calvin cycle, like Rubisco, can become denatured, reducing the rate of photosynthesis. Water availability. Water stress can reduce the rate of photosynthesis. Stomatal closure under water stress limits the amount of CO entering the leaf, thus limiting photosynthesis. Chlorophyll and other pigments. The efficiency of light absorption depends on the amount and type of pigments. Chlorophyll A is a primary pigment, but accessory pigments such as chlorophyll B and carotenoids help in the absorption of additional light wavelengths. Photosynthesis and energy efficiency. The process of photosynthesis is relatively inefficient in terms of energy conversion. Only about 1-2% of the sun's energy is converted into chemical energy. This efficiency can be influenced by various environmental and biological factors, such as leaf structure, chloroplast number, and pigment composition. Previous year CSIR NET questions. CSIR NET June 2019. Question. In the light reactions of photosynthesis, the excited electron from chlorophyll ultimately reduces which of the following? A NADP+. B rub C oxygen D glucose. Answer. A NADP+. The excited electrons are ultimately transferred to NADP+, reducing it to NADPH. CSIR NET December 2020. Question. Which of the following processes does not occur during the light-dependent reactions of photosynthesis? A splitting of water. B formation of ATP, C reduction of NADP+ to NADPH, D formation of glucose. Answer. D formation of glucose. Glucose formation occurs during the light-independent reactions, i.e., the Calvin cycle. CSIR NET June 2017. Question. The enzyme Rubisco plays a key role in which stage of photosynthesis? A light-dependent reactions. B carbon fixation, C electron transport chain, D NADP+ reduction. Answer. B carbon fixation. Rubisco is responsible for fixing CO into RUB during the Calvin cycle. This lecture on photosynthesis provides a comprehensive overview with a strong focus on exam relevance, ensuring you grasp the core concepts crucial for the CSIR NET JRF Life Sciences exam. The inclusion of previous year questions further strengthens the understanding and helps in exam preparation. You said. Light harvesting complexes. ChatGPT said. Here's an elaborate lecture on light harvesting complexes, tailored for the CSIR NET JRF Life Sciences exam. This lecture focuses on the key concepts, important points, and exam-oriented details from base to advanced level, without wasting space, and includes 2 to 3 previous year questions with answers. Light harvesting complexes (LHCs) in photosynthesis. Light harvesting complexes (LHCs) are essential components of the photosynthetic machinery in plants, algae, and cyanobacteria. Their primary role is to capture light energy and funnel it to the reaction center where the energy is used for the photochemical reactions of photosynthesis. The LHCs are composed of proteins and pigments that work together to maximize light absorption. LHCs are part of a larger structure called the photosystem, either photosystem I (PSI) or photosystem II (PSII), which is involved in the light-dependent reactions of photosynthesis. The pigments in the LHCs absorb light energy, and through resonance energy transfer, this energy is passed on to the reaction center where it drives the production of ATP and NADPH. Structure of light harvesting complexes pigments. The light harvesting complexes contain several pigments, including chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A is the primary pigment that absorbs light, while chlorophyll B and carotenoids act as accessory pigments, broadening the spectrum of light absorption. Proteins. LHCs consist of membrane-bound proteins that form the structural framework, allowing the pigments to be embedded in the thylakoid membrane. These proteins help in organizing the pigments and facilitating the transfer of absorbed light energy. Arrangement. In photosystem II, the LHC2 light harvesting complex 2 absorbs light primarily in the red and blue regions of the spectrum. In photosystem I, the LHCI light harvesting complex I absorbs light, transferring energy to the PSI. Function of light harvesting complex is absorption of light energy. The LHCs are responsible for capturing light and exciting electrons in the pigments. This process is called photoexcitation. The absorbed light energy is transferred through a series of pigment protein complexes to the reaction center of the respective photosystem. Resonance energy transfer. When light is absorbed by the pigments in the LHCs, the energy excites the electrons to a higher energy state. The energy is passed through resonance energy transfer from one pigment molecule to another until it reaches the reaction center. In PSII, the energy is transferred to the reaction center where it leads to the photolysis of water and the production of oxygen. Energy transfer to reaction centers. In photosystem 2, the energy absorbed by LHC2 is transferred to the P680 reaction center, leading to the excitation of electrons that initiate the electron transport chain. In photosystem I, LHCI transfers the energy to the P700 reaction center, which is involved in the reduction of NADP+ to NADPH. Efficient light absorption. In photosystem 2, the energy absorbed by LHC2 is transferred to the P680 reaction center, leading to the excitation of electrons that initiate the electron transport chain. In photosystem I, LHCI transfers the energy to the P700 reaction center, which is involved in the reduction of NADP+ to NADPH. Efficient light absorption. The LHCs are strategically arranged to ensure that they can capture light over a wide spectrum and maximize the absorption of available light energy, especially under low light conditions. Types of light harvesting complexes. LHCI, light harvesting complex 2. LHCI is the main light harvesting complex associated with photosystem 2. It is composed of chlorophyll and chlorophyll B as well as carotenoids. LHCI absorbs light in the blue 430-450 nanometers and red 640-680 nanometers regions of the spectrum. The energy absorbed is transferred to P680 in PSII. LHCI, light harvesting complex I. LHCI is associated with photosystem I and primarily absorbs light in the red region of the spectrum 700-740 nanometers. It is composed mainly of chlorophyll and is involved in transferring absorbed energy to P700 in PSI. Phycobilisms in cyanobacteria and red algae. In cyanobacteria and red algae, the LHC is replaced by a complex called the phycobilism, which contains phycobilins that absorb light in the green and yellow regions of the spectrum. Regulation of light harvesting complexes. The efficiency of photosynthesis is regulated by the state transition mechanism, which allows the plant to adapt to changing light conditions. When the light conditions change, the LHC proteins in the thylakoid membrane can move between the photosystem I and photosystem 2 to ensure optimal light harvesting and energy distribution. This mechanism is crucial for maintaining the balance between the two photosystems and preventing photoinhibition, damage caused by excess light. Importance of light harvesting complexes in photosynthesis. Enhance light utilization. LHCs increase the efficiency of photosynthesis by capturing light energy across a broad spectrum of light, including wavelengths that chlorophyll alone cannot absorb efficiently. Optimization of energy transfer. By transferring energy effectively from the light harvesting pigments to the reaction centers, LHCs ensure that the photosynthetic process runs smoothly, leading to the efficient production of ATP and NADPH, which are crucial for the Calvin cycle. Adaptation to light conditions. LHCs play an important role in the plant's ability to adapt to varying light conditions. They allow the plant to maximize light capture under low light conditions and adjust to avoid damage when light is too intense. Previous year CSIR net questions. CSIR net June 2018. Question: which of the following is a major component of the light harvesting complex 2 LHC2? A. Chlorophyll. A. B. Chlorophyll B. C. Carotenoids. D. Phycobilins. Answer: B. Chlorophyll B. Chlorophyll B is a major component of LHC2, helping in light absorption and energy transfer. CSIR net December 2017. Question: in the light-dependent reactions of photosynthesis, the main role of the light harvesting complex is 2. A. Splitting of water. B. Fixation of carbon dioxide. C. Capture light energy and transfer it to the reaction center. D. Production of NADPH. Answer: C. Capture light energy and transfer it to the reaction center. LHCs capture light energy and funnel it to the reaction centers, where the energy is used for photochemical reactions. CSIR net June 2016. Question: the primary function of light harvesting complex 2 LHC2 is 2. A. Split water molecules. B. Transfer energy to photosystem I. C. Absorb light and transfer energy to photosystem 2. D. Produce oxygen. Answer: C. Absorb light and transfer energy to photosystem 2. LHC2 absorbs light and transfers the energy to the reaction center of photosystem 2. This lecture provides a detailed and exam-focused understanding of light harvesting complexes, with a strong emphasis on key concepts and previous year questions to help in CSIR net JRF life sciences preparation. It covers all essential aspects, including structure, function, types, regulation, and significance, to ensure you are thoroughly prepared for the exam. You said. Mechanisms of electron transport. ChatGPT said. Here's an elaborate lecture on mechanisms of electron transport, tailored for the CSIR net JRF life sciences exam. This lecture is designed to focus on key concepts, important points, and exam-oriented details from base to advanced level, without wasting space, and includes 2 to 3 previous year questions with answers. Mechanisms of electron transport in photosynthesis. The electron transport chain, etc., is a critical component of the light-dependent reactions of photosynthesis. It is responsible for the transfer of high-energy electrons from water molecules to NADP+, thereby generating NADPH and ATP, the key energy carriers used in the Calvin cycle for carbon fixation. The electron transport mechanism is highly efficient and involves a series of protein complexes and mobile electron carriers embedded in the thylakoid membrane of the chloroplasts. Overview of the photosynthetic electron transport chain. Photosystem 2 PSII. The electron transport process begins at photosystem 2 PSII, where light energy is absorbed by chlorophyll and accessory pigments. This energy excites electrons, which are then transferred to a primary electron acceptor. The P680 reaction center of PSII is responsible for splitting water molecules, a process known as photolysis. This releases oxygen O2 and protons H+. The electrons released from water flow through the electron transport chain. Plastoquinone PQ. The excited electrons from PSII are transferred to plastoquinone PQ, a lipid-soluble electron carrier. The electron transport process begins at photosystem 2 PSII, where light energy is absorbed by chlorophyll and accessory pigments. This energy excites electrons, which are then transferred to a primary electron acceptor. The P680 reaction center of PSII is responsible for splitting water molecules, a process known as photolysis. This releases oxygen O2 and protons H+. The electrons released from water flow through the electron transport chain. Plastoquinone PQ. The excited electrons from PSII are transferred to plastoquinone PQ, a lipid-soluble electron carrier. PQ transports electrons from PSII to cytochrome B6F complex and simultaneously carries protons into the thylakoid lumen, contributing to the formation of a proton gradient. Cytochrome B6F complex. The cytochrome B6F complex is a large multi-subunit enzyme complex that plays a crucial role in the electron transport chain. It receives electrons from plastoquinone PQ and transfers them to plastocyanin PC. The complex also pumps protons into the thylakoid lumen, enhancing the proton gradient, which is used to drive ATP synthesis through ATP synthase. Plastocyanin PC. Plastocyanin PC is a small copper-containing protein that carries electrons from the cytochrome B6F complex to photosystem I PSI. Plastocyanin plays a crucial role in maintaining the continuity of the electron flow between PSII and PSI. Photosystem I PSI. Electrons are transferred from plastocyanin to the P700 reaction center in photosystem I. The energy from light excites the electrons in PSI, and these high-energy electrons are transferred to a primary electron acceptor. PSI is responsible for producing NADPH by transferring electrons to NADP+, which is reduced to NADPH. Ferredoxin FD. The electrons from PSI are transferred to ferredoxin FD, a small iron sulfur protein. Ferredoxin plays a key role in transferring electrons to NADP+ reductase for the final step of NADPH production. NADP+ reductase. The enzyme NADP+ reductase catalyzes the reduction of NADP+ to NADPH by utilizing the high-energy electrons from ferredoxin. This NADPH is essential for the Calvin cycle, where it provides the reducing power required for carbon fixation. Key steps in the electron transport process. Absorption of light energy. Light energy is absorbed by photosynthetic pigments, chlorophyll and B, carotenoids in photosystem 2 PSII. The energy is transferred to the P680 reaction center, which excites electrons to a higher energy level. Water splitting photolysis. The excited electrons are transferred from P680 to a primary electron acceptor, and water molecules are split to replace the lost electrons. This produces oxygen O2 and protons H+. Electron transfer to plastoquinone. The excited electrons from PSII are transferred to plastoquinone PQ, which carries the electrons to the cytochrome B6F complex. Proton pumping and proton gradient formation. As electrons move through the cytochrome B6F complex, protons are pumped from the stroma into the thylakoid lumen, establishing a proton gradient across the thylakoid membrane.